Hello my dear friends, welcome to Live Prep. In the previous lecture, we studied about the basic introduction regarding the body fluids and circulation, and then we have started about the blood. So in the blood we studied about the two components, and one is the fluid matrix that is called as plasma. So within the blood, the fluid matrix is called as fluid matrix. The matrix is fluid in nature, so that's why it is called fluid matrix. It is called as plasma. And regarding the plasma, we studied that 55% of blood, 55% of blood is the plasma. And within the plasma, 90 to 92% water is present, 6 to 8% proteins are present. In the proteins also, we studied about the fibrinogens, albumins, globulins. Not only this, some other proteins required for blood clotting are also present in the blood plasma. And then we studied that even some minerals are also present, some nutrients are present, waste materials are present, gases are present, and even some hormones are also present. Likewise, so many different types of components are present in the blood plasma. So up to this we studied in the previous lecture, and now we are focusing on the other part. What is the other part for the blood? One part is fluid matrix called as plasma. So other part is formed elements. Formed elements. What are these formed elements? These are nothing but cells. Formed elements are nothing but cells. And these cells, again, they are of how many types? They are three types. They are three types. What are these three types of the cells? Now let us see one by one. What are these three types of the cells? First one are called as RBC. What are these RBCs? They are red blood cells. Red blood cells, you can also call them as red blood corpuscles. Anything you can say, red blood cells. And here they are also called as erythrocytes. 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 Erythro means red, cyte means cell. So they are red cells. They are red in color. That's why they are called as erythrocytes. And the next type are called as WBCs. WBCs. Okay. Plural I am writing. Small s. WBCs. What are these WBCs? They are white blood cells. They are white blood cells. And these are also called as leukocytes. Leukocytes. The term leuco means colorless. Leuco means color. They are not having any color. That's why we are saying them as white blood cells. But actually they are colorless. Where we studied about the term leuco, when we studied about the cell chapter, there we studied about the plastids in the plants and euglenoids. So those plastid classifications, so there we studied that if pigments are not present in the plastids, they are called leucoplasts, colorless plastids. There we used the term leuco. Here also you are using the term leuco. Yes or no? Leuco means colorless. They don't have any colored pigments. That's why they are colorless. And apart from this, there is another one that is called as platelets. Platelets, and these are also called as thrombocytes. These are also called as thrombocytes. Yes or no? So this is what the general basic information that we are studying here. Now with respect to RBCs, now we will go point by point here. Okay. So first we will focus on the RBCs, then we will come to the WBCs. So regarding the RBCs, already some general information we studied in the different other chapters. Now first we will see here. Formed elements are the cells. So if 55%, is the fluid matrix plasma, what is the percentage for the formed elements? It is 45%. 55 plus 45, so it is 100%. So in this way, formed elements will account for 45% of the blood. So that's what we are writing here. And now here three types of cells: erythrocytes, RBCs, leukocytes, WBCs, thrombocytes are the platelets. And in general, what is this hematopoiesis? Hematopoiesis is the production of these blood cells. Production of these blood cells is called as hematopoiesis in simple. Specifically, if you are talking about the erythrocytes, erythropoiesis, leukopoiesis, thrombocytes, thrombopoiesis. Separately, you can use it. For all the blood cells together you are talking, it is called hematopoiesis. This is the term. The production of these blood cells is called as hematopoiesis. Clear? Next one. Erythrocytes, leukocytes and platelets are collectively called as the formed elements. So these three are combinedly called as formed elements. That means these are the cellular part, the cells that are present in the fluid connective tissue blood, and they constitute nearly 45% of the blood. That's what we have studied here. And now we are specifically talking about the RBCs. First we will study the information about RBC, then we will go with NCERT content for RBCs. What are the important points? RBCs are also called as red blood cells or erythrocytes. And what is the normal count? What is the normal count for these RBCs? Now we are going point by point, so observe very carefully here. So first we are talking about RBCs. What is the normal count here? 5 to 5.5 million RBCs per millimeter cube. Per millimeter cube. So instead of writing per millimeter cube, you can write this as millimeter inverse cube. Okay, so millimeter power minus 3, like this also you can write, meaning is same only. That means in 1 millimeter cube volume, that means in 1 cubic millimeter, these many RBCs are present. If you compare about all the cells, RBCs, WBCs, platelets, RBCs are the most abundant, because for all the three, let us say WBC I will write here, platelets I will write here. It will be easy for you to compare. Here we are taking for millimeter cube only. All the three we are comparing for 1 millimeter cube. But here how many are present WBCs? Just 6,000 to 8,000. But whereas about this particular platelets, if you see 1,50,000 to 3,50,000, these many cells are present. But here 5 to 5.5 million. Suppose you are writing in normal terminology for the RBC, what you will write here? 50 lakhs. 5 million means 50 lakhs to 5.5 million means 55 lakhs. This is the number. You just see it. Here this is 1.5 lakh to 3.5 lakh. That's it. But these are in just thousands, 6 to 8,000. That's it. But these are 50 lakhs to 55 lakhs. That means 5 million to 5.5 million. Now you just see who are more in number. RBCs are the most abundant cells within the blood. The term most abundant they have used in the NCERT. So please understand this. This is what the general comparison is. So number wise, if you say, RBCs are the most abundant cells that are present in the human blood. This information we are studying for human only. Please understand. This is about the normal count. Next one regarding the production. Production of RBCs is specifically called as erythropoiesis. And where exactly this erythropoiesis will begin, observe carefully. In the embryonic stages, if you are talking, in the embryo condition, if you are talking, initially RBC production or erythropoiesis begins with yolk sac. Initially it is occurring at yolk sac, and then it will go to the spleen. That means after the yolk sac, now the location is changed, and now it will start producing from the spleen, and after that again it will change to liver. That means RBC production will occur from the liver. And at the age of 7 months, and now it is starting from the approximately at the age of 7 months, it is starting from the bone marrow. So from then onwards, after the birth and every time, it will be bone marrow only. So that is the reason why this we are observing only in the embryonic stages. Bone marrow also we are observing, but after 7 months of the embryo. So from then onwards, every time it is from the bone marrow only. So in this way, in the NCERT they are talking only about the bone marrow, because they are saying about adults. Adult means only the bone marrow. So in this way, bone marrow is the location at which this RBC production is occurring. Not only RBC, WBCs are also, most of these WBCs are also produced from bone marrow only, and even the platelets are also. All these are mainly produced from bone marrow only. That's why hematopoiesis is occurring at the bone marrow from 7th month onwards. This is what the general information. But please remember, in the embryonic stages, these are also for temporarily for few days or few months, these are also involved in the hematopoiesis, blood production. That's it. But please remember, bone marrow, mainly which bone marrow? Long bones. Mainly the long bones, they are involved in this blood production. So in the case of long bones, I will draw one rough diagram. But please remember anyhow, regarding the bone, general structure of the bone, since the tissues chapter is deleted, generally we are not studying about this one. So generally it is present like this. The bone is generally present like this. So now here for this, these are the ends. And please remember, inside this bone, now you will get the bone marrow. So at the ends of these long bones, you will find red bone marrow. And here at this spot, you will find yellow bone marrow. Okay. And now which one is actively involving in the blood production? Blood means blood cells production. It is the red bone marrow. So it is the red bone marrow present at the ends of long bones like this, thigh bones, and then these hand bones, and then in some cases ribs and even the pelvic bones are also involved in the blood production. Okay, but mainly major production is occurring from the long bones. For the long bones you will see these ends, and at these ends on the inner side you will find the red bone marrow. From this red bone marrow actually this blood cells production will occur. So RBC production is also occurring from this one. Clear? So in this way, in simple, if you are talking about the location, so it is produced from the red bone marrow. That's it. That's it. What about the location of this production? So that from that they are released into the blood. They are produced there and then they are released into the blood. This is what. And then regarding the shape, this is very important concept. You need to understand these particular points. So regarding the shape, if you are talking, now let us see here. In the case of fishes, amphibians, reptiles, and even the birds. In all these cases, in general, if you are talking, in all these cases, in the case of RBC, nucleus is present. In the case of RBC, nucleus is present. Please observe carefully. In these organisms, RBC has nucleus. Since nucleus is present, now it will be biconvex. This is the convex shape. This is also convex shape. So it is a biconvex in these particular organisms. So here in the mammals, here we are using a word called many mammals. Why you are saying many mammals? Because exceptions are there. Already in the cell chapter, when we studied nucleus, there we studied camel and llama. Camel and llama, they will come under this particular one. Even though these are mammals, again they are having nucleus, so they will have biconvex shaped RBCs. But in the case of many mammals, except this remaining, here what you are saying, here if you are observing like this, it is biconcave, and it is round. Okay, so if you are observing like this, it is round, and here if you are observing, it is biconcave. So here nucleus is absent, and here it is biconcave shape. This is what the general information. But here we are talking about the human. So here we will get about the human. So with respect to human, if you say mature RBC, please remember, when RBC are initially produced, initial RBC is immature RBC. When it is immature, nucleus is there. But during maturation, what happens? During maturation, the nucleus will be lost. Why it is losing the nucleus? And I already told about this. It is not just nucleus. Every student is remembering about nucleus loss. It is not just nucleus, other cell organelles are also lost. Nucleus and other cell organelles are also lost. Why they are losing? Why these cells are losing this nucleus and other organelles? Because anyhow, in the mature RBC, for a particular duration of 120 days, it is surviving. After that, anyhow, it is undergoing degradation. And that too, these RBC do not undergo division to form next generation RBC. So these RBCs cannot divide further. New RBC are again produced from the bone marrow. So that's why, anyhow, their life is for only particular few days. And for the survival of these many days, whatever the proteins that are required, it will produce all the proteins. Once these proteins are present, it can survive for those many days. That means technically that nucleus and other organelles are not required. So that's why it is losing it. And why it is losing? Upon losing this, a space is created. In that space, it can accommodate more and more hemoglobin. Please understand this. It is losing the nucleus and other organelles so that it can accommodate more and more hemoglobin. If more hemoglobin is accommodated, now it can transport more oxygen. Already in the transport of gases, in the previous chapter already we studied this. Hemoglobin is binding with oxygen and forming oxyhemoglobin. Hemoglobin is binding with CO2 and forming carbaminohemoglobin. So hemoglobin is important for transport of oxygen and CO2. So for the transport of those gases, hemoglobin is required. If these organelles are removed, more hemoglobin can be accommodated. So more these gases can be more efficiently they are transported. For that purpose, it is losing the nucleus and other organelles. Understood or not? That's why mature RBCs do not contain the nucleus. But still in the cell chapter they asked a question: do you consider them as living? Yes, we should consider them as living cells. Even though they are losing the nucleus, still they are considered as living cells. They are working, they are performing so many functions. They are not dead cells, they are working cells. So they are still considered as living cells only. Understood or not? This is the information. So here we are observing biconcave. So in the cell chapter, in the initial part, we studied about shapes of cells. There we studied about they are round. They are round and biconcave. So here superficially, if you see, here they are round like this, but here they are like this. So that's why round and then biconcave. So that's why they are round biconcave in the case of human, not just for human, for many mammals. But for camel and llama, since nucleus is present, they are having the biconvex shape. Clear? With this point is completed. Now we need to go to the next point. But before going to the next point, here itself I am telling you the comparison. For the human RBC, mature RBC does not contain nucleus and it is having round and biconcave shape. Anyhow, frog is there in our syllabus, right? Frog will come under amphibian. So, mature RBC of frog is having nucleus. So, it will have biconvex shape. Please understand this. So, when we study about the frog, again this point will come. So, we will interlink this content. So, please understand this one. So, this is some general basic information regarding this RBC shape. Next one is regarding the color. What is the color of this RBC? Red. Red blood cells. What is the color? They are red in color. Why they are having red color? Because they are having red color pigment that is called as hemoglobin. Hemoglobin is present. So because of the hemoglobin, cells are appearing red. And not just the cell, even blood is also appearing red. Yes or no? What is the color of the blood? Now it is red in color due to the presence of red blood cells. Why red blood cells are red in color? Because of the hemoglobin. So because of hemoglobin, RBC are getting red color and even the entire blood is appearing in red color. Please observe this one. Very, very important one. And then what is the hemoglobin role? Already in the previous chapter, breathing and exchange of gases. Already we studied about this. What we studied? So 100 ml blood, 100 ml blood will carry 20 ml oxygen, we are saying. What is the calculation we took there? So 100 ml blood is having approximately 12 to 16 grams hemoglobin. Yes or no? If you are taking 15 grams average, it is carrying 20 ml of oxygen. How you are calculating this? 1 gram of hemoglobin, approximately it is carrying 1.3 ml oxygen. This is the calculation. When you are multiplying with 15, you will get 20 point something. For the round figure, we are taking 20 ml. Sometimes you can take 21 also, no problem. 20.8 somewhere you will get it. Any value you can take, no problem. This is what the average calculation. So in this way, hemoglobin is very, very important for the transport of these gases. That is what the role. It is involving in both transport of oxygen and also involving in transport of CO2. When oxygen is combined with hemoglobin, it will form oxyhemoglobin. And when CO2 is combined with hemoglobin, it is forming carbaminohemoglobin. All the points very clearly we studied in previous chapter. Next one, lifespan. What is the average lifespan of this particular one? Average lifespan is 120 days. What happens after 120 days? Generally what happens? They are circulated in the blood circulation. So now this blood is also even going to an organ that is called as a spleen. Spleen. Okay, so just above the stomach it will be present on this left upper part. Spleen is present. Now it is going to the spleen and the spleen is also getting this blood circulation. And now when this blood is passing through the spleen, now RBCs are also present in this blood. So those cells whose life is coming approximately to 120 days. Now what happens? Here there are some cells, macrophage cells are present, okay, and those cells, there are some cells and those cells, they will collect these particular RBCs and then they will start the breakdown of these cells. And these cells, they will be broken down and then that hemoglobin will be collected and again it can be reused. Okay. So in this way, since the spleen is removing the damaged or the dead RBCs or the whose lifespan is completed. So these RBCs will be removed by the spleen. That's why spleen is considered as a graveyard. So once we die, ultimately they will take us to the graveyard. Same way, so these RBCs, finally they are removed at the place called as spleen. That's why spleen is considered as graveyard of RBCs. Graveyard of RBCs. In the NCERT, very clearly they gave this. But in higher classes, you will study that anyhow spleen is there. Along with the spleen, even liver is also contributing to some extent. In the case of liver also, some RBCs will be there will be same process will be done. So those whose life is completed, now some of these RBCs will be collected there and they will be damaged there also. So in this way, liver is also involving in this process, but the major one is a spleen. That's why spleen is considered as graveyard of RBCs. But in the option they are giving, one is spleen, other one is liver. You should go for spleen only because very clearly mentioned in NCERT. Clear? This is what the basic information regarding the RBCs. Now let us go through NCERT content. What they have given here. Erythrocytes are also called as red blood cells. They are the most abundant of all the cells in blood. RBC, WBC, platelets. If you are comparing all these are the most abundant because their number is very huge. Next one. A healthy adult man has on an average 5 million to 5 million RBCs per cubic millimeter of the blood. So these many are present. 5 millions means what? 50 lakhs. 5.5 million means what? 55 lakhs. These many are present. RBCs are formed in the red bone marrow in the adults. In the case of adults, they are produced from red bone marrow of long bones mainly. Next one. RBCs are devoid of the nucleus. Devoid of nucleus means what? Nucleus is absent. In the case of RBCs, nucleus is absent in most of the mammals. Why they are saying most? Because camel and llama, they are mammals, but still they are having nucleus in their RBCs. That's why for the most of the mammals, mature RBCs do not contain the nucleus. That is what the meaning is. And are biconcave. If nucleus is present, it will be biconvex. Since nucleus is not present, they will become biconcave shape, and this biconcave shape will help in easy passage of these RBCs through small capillaries also. There is an advantage here also. Okay, next one. They have red colored iron containing complex protein that is called as hemoglobin. Very clearly we studied about this in the previous chapter. Hence the color. That's why they are getting the red color. So that's why RBC is getting the red color due to presence of hemoglobin. And name of the cells. Name of the cells is red blood cells due to presence of this red colored pigment called hemoglobin. Next one. A healthy individual has 15 to 16 grams of hemoglobin in every 100 ml of blood. 100 ml blood he is having approximately 12 to 16 grams of hemoglobin. And these molecules play significant role in the transport of respiratory gases like oxygen and CO2. Already in the previous chapter we studied. RBCs have an average lifespan of 120 days, and after which they are destroyed, they are damaged, they are lysed in the spleen. That's why spleen is considered as graveyard of RBCs. This is what the information they give. General, very simple information, but each and every point is important for examination. This is the general information with respect to the RBCs. Now we will see the similar information with respect to the WBCs. With respect to WBCs, now let us see here. With respect to WBCs, first they are talking about the normal count. Already we studied about the normal count for WBCs. What is the normal count for WBCs? Normally in the range of 6,000 to 8,000 cells per millimeter cube. For cubic millimeter they are present. This is the normal range. WBC means all the types of WBC, all the types. Now I will tell you types also. All of them combinedly, this is the average value. Okay. So in this way, this is the normal count. So that means these are the least abundant. Most abundant are RBCs. Least abundant are WBCs. Medium abundant are platelets. Okay. Next one. And production. And the production of this particular WBCs is called as leukopoiesis. Yes or no? Erythropoiesis for RBC production, leukopoiesis for WBC production, and thrombopoiesis for platelet production. So that's why production of this particular one is called as leukopoiesis, and these are also produced from the bone marrow generally. Okay, and specifically with respect to lymphocytes, some lymphoid organs are involved, but for other types of WBCs again from the bone marrow only they are produced, and from there they are produced and released into the blood. This is what they are saying. And regarding the shape, please remember in the cell chapter already we studied about the shape. Shape of WBC is amoeboid. In the cell chapter we studied about the shape of this particular one. Shape of RBC is round and biconcave for human. And what about this particular WBC? It is amoeboid. That means it is irregular. Irregular. Any type of WBC you just take it, it is irregular. So randomly you just draw the diagram. This is the shape. The shape of WBC is irregular. It is amoeboidal. Very, very important point. So please remember this. Next one. Color. They don't have hemoglobin. Hemoglobin is absent. And they are not having any other pigment containing molecule. So that's why they are not having any color. That's why they are called leukocytes, colorless, and you can also call them as white blood cells. So that's why they are not having any color. Nice. Next one, hemoglobin. It is absent. It is present only in the RBC. It is not present in WBC, not present in platelets also. Next one, what is the lifespan here? Generally, lifespan of WBC, generally if you are talking, it may be for few hours to few days, that's it. Few hours to very few days, but within the WBCs, anyhow we will study the classification. Now I will tell you the classification of WBC, observe carefully. Within the WBC, there are two major groups are present. They are called as granulocytes and agranulocytes. Granulocytes and agranulocytes. Granulocytes, granule containing cells. Cytes means cells, granule containing cells. But agranulocytes, these cells do not contain any granules. And they are having some granules inside these cells. And these granules are having different abilities. And some cells are having the granules, and these granules can easily bind with acidic dyes. So that's why these are called as acidophils. These are called as acidophils. That means acid fill loving. That means these granules present in the cells can easily interact with acidic dyes. That's why they are called acidophils. And these are also called as eosinophils. Eosin is a dye. They can easily bind with eosin. That's why they are called eosinophils, or simply called as eosinophils. They are simply called as eosinophils, but actually it is the eosin dye. So they are called as eosinophils or eosinophils. Eosinophils or acidophils. And the other type of cells, some cells are having the granules. They are readily binding with basic stains. That's why they are called as basophils. So basic stains interaction. So basic loving. That means they can easily bind with basic dyes. That's why they are called basophils. And some are binding with the neutral dyes. That's why they are the neutrophils. Neutrophils. So in this way, WBCs are two groups, granulocytes and agranulocytes. Within the granulocytes, again, three types are there: acidophils, basophils, neutrophils. These will bind with acidic dyes, these will bind with basic dyes, and these are readily binding with neutral dyes. That's what. And regarding this agranulocytes, two types are there. They are lymphocytes and monocytes. Lymphocytes and monocytes. Please remember here. And now we are talking about the lifespan. In general, if you are talking few hours to few days only is the lifespan. But within these lymphocytes, you will study about two types again. B lymphocytes, T lymphocytes. Okay. Within this again, you will study about plasma B cell, and then memory B cell. Plasma B cell and memory B cell. So here also, generally we are not using the term called as a plasma, but here you can see this activated T cell, and also there is a memory T cell. I don't want to give more information here because unnecessarily you will be confused here. So one is the readily activated T cell and other one is the memory T cell. So now my main focus is memory T cell. My main focus is memory cells. So within the B cells there is a group. Within the B cells there is a group called as memory B cells. Within the T cells also there is a group called as memory B cells. So these memory B cells can survive for several years. They can survive for several years. And regarding these memory B cells, memory T cells, you will study more details in health and diseases chapter. Here I don't want to add more information. Here at least up to B and T cells, you please remember that is enough. Okay. So here these memory B and memory T cells, they can survive for longer duration. It may be up to several years also. They are very, very important for immune function. They are protecting our body. Okay. So whenever a pathogen is coming into our body, these cells will recognize them. Once they are recognizing, they will divide into one immediately acting cell, one for future use. That future use is the memory cells. And these memory cells have the information in their in their system, so that in future if the same pathogen is coming, immediately they will start acting on it. For that quick immediate action, these memory cells are very important. That's why they are surviving for several years. Only this type of cells can survive for longer duration, but remaining other types, they are having very less lifespan. This is what the point that you need to understand under the lifespan. Next one is regarding types of leukocytes. Just now we studied granulocytes are three types. Eosinophils are eosinophils, basophils, neutrophils. Agranulocytes are two types, lymphocytes, monocytes. And here what is the next important information? This is how they are appearing. And what is the other important information regarding the shape of nucleus? We need to observe very carefully. Shape of the overall cell, it is irregular. Okay. Shape of the overall cell, it is irregular. Okay. Now this point I am removing. Now I will draw the rough diagram. So please observe carefully here. So overall rough diagram if you are drawing, for the cell if you are drawing, they are irregular. They are irregular here. All these are irregular. For the shape of the cell it is irregular. But if you are observing the shape of the nucleus, generally these eosinophils are having two lobes. This is one lobe and this is other lobe. This is one lobe, this is other lobe. So it is bilobed. Bilobed nucleus. Bilobed nucleus. For the cell, shape of the cell it is irregular. For the nucleus there are some fixed shapes are present. So eosinophils generally they are having bilobed nucleus. In the case of basophils, generally they are having trilobed nucleus, and sometimes they can also have bilobed, but generally they are having a trilobed. Tri means three lobes, trilobed nucleus. In the case of neutrophils, generally they are having multilobed. Generally they are having multilobed like this. So more than like this. So here it is generally multilobed nucleus. I am giving the general ones. Sometimes some other forms also you can see. Generally it is eosinophils are bilobed, basophils are trilobed, and neutrophils are multilobed. Nice. Coming to the lymphocytes. In the case of lymphocytes, generally it is having spherical shaped nucleus. Lymphocytes, whether B lymphocyte, T lymphocyte, whatever it is, lymphocytes they are having spherical shaped nucleus. But in the case of monocytes, they are having bean shaped or kidney shaped. This is the spherical shaped, and here it is the kidney or bean shaped or kidney shaped nucleus is present. So in this way, cell shape of the cell if you are observing, it is irregular for WBC, any WBC. But if you are talking about the shape of the individual nucleus, yes, it is having a specific shape here. So this is what the information that we are studying here. And that too, this neutrophils within this nucleus, there is a concept called as bar body concept that we will study in future, but remember the name here. So drumstick arrangement, we are observing generally in the neutrophils of females. In the nucleus of females, human females, we are observing a drumstick arrangement. Generally what we are observing, suppose this is the nucleus, I am making a spherical nucleus. Inside this you can see some stick like arrangement that is attached to inner side of nucleus, and this is like a drumstick. Okay, the stick, it is like a drumstick, so that's why it is called a drumstick arrangement, and this is related to bar body concept. It is related to bar body concept, and this concept I will explain you in Principles of Inheritance and Variation, that is a class 12th chapter. In the class 12th chapter, Principles of Inheritance and Variation, we will study about bar body. This bar body is nothing but X chromosome. In human females only you are observing. In the males you will never see this. In males X is present, but that is not present like this. Please observe this one. This bar body concept is something different, and that was given in the QR code content of Principles of Inheritance and Variation. So I will explain in Principles of Inheritance and Variation. Clear? Please remember this. These bar bodies are seen in, bar bodies are present in all the cells of human female. All the cells of human female are having bar body, but they are clearly seen as a drumstick in the neutrophil cells. This point is something not mentioned in NCERT, but my suggestion is please better try to remember this particular information. Clear? Now let us see shapes of the nucleus. So different types of WBCs are having different shapes. Just now we have studied about this one. This is what the same information I have given in the form of table. And next we are talking about the general information. What is the general information? What is the significant point for these particular types of cells? This is what the information we are studying. So directly with the help of this diagram itself, I will try to explain it. Observe carefully. First we will see on this eosinophils or acidophils. So generally they are related with some allergic reactions. In some allergic reactions, the number of eosinophils will increase, so that you can call this as eosinophilia. Eosinophilia. Please remember this. So these eosinophils or acidophils are generally related with some allergic reactions, and even some parasitic infections also. Even in some parasitic infections like helminthic parasites infections, these are involved, so their number will increase. So this is what the relation. Two important points related with allergic reactions and also some types of parasitic infections. This is one important point you need to remember for acidophils. And now we are talking about the basophils. And the basophils, generally they are releasing some compounds like heparin, histamine, and serotonin. They are having different functions, but mainly these basophils are releasing histamine and serotonin in relation with inflammation. So they are involving in inflammation formation. Inflammation means it is the swelling, redness, heat production. So many characteristics are present for inflammation. So basophils are releasing some chemicals called histamines and serotonins, and mainly they are involving in inflammatory process. They are causing some inflammations. This is one important point. And basophils are also releasing another chemical that is called as heparin. Very, very important one. Heparin is called as anticoagulant. Heparin is called as anticoagulant. Heparin is anticoagulant. It is anticoagulant. Please observe carefully. What is this anticoagulant? Coagulation of the blood clotting. Coagulation is the clotting of the blood. But what the heparin function? Anticoagulant, anti-clotting. Anti-clotting means what? Heparin will prevent clotting of the blood. This is very, very important point. You please remember. Regarding this function, once again we will study under excretion chapter. There we will study about hemodialysis, artificial kidney concept. There also I will explain the concept once again. So please observe carefully. What is the function of heparin? See, when blood is flowing in the blood vessels, it should not clot. Suppose the blood is clotting, then how it will flow? It should be in the liquid form. It should be continuously flowing. So that means clotting should not occur. So the blood that is flowing in the blood vessels, it should not clot. So that's why this heparin is very, very important. So in this way, this heparin will make sure that blood is not coagulating when it is flowing in the blood vessels. Yes or no? Where blood clotting should occur? If there is a damage to the blood vessel, at that damaged portion only clotting should occur. But the flowing blood should not undergo coagulation or clotting. Yes or no? So that's why this heparin present in the blood will make sure that the flowing blood is not undergoing clotting, not undergoing coagulation. So that's why it is very, very, very important one. Clear? Next one, neutrophils. Neutrophils are phagocytic cells. Neutrophils are phagocytic cells, and these are the most abundant, and even the monocytes are also phagocytic. What is phagocytic? In the cell chapter I told you about endocytosis and exocytosis concept, right? Endocytosis, exocytosis concepts I told you. In the endocytosis we studied about phagocytosis and pinocytosis. Phagocytosis and pinocytosis. Phagocytosis is called cell eating. So there what you are saying, suppose this is a cell and this is suppose a food material or a pathogen. Now what will happen here? Now this will form a loop around this. So that now finally what happens? Now this food material is coming here like this, and then finally what happens here? Now this is making like this. Yes or no? In this way that solid food or pathogen is taken into the cell. So taking like this, a solid food or pathogen taking like this into the cell is called phagocytosis. Same way Same way, this neutrophil and neutrophil and monocytes, they are involving in phagocytic process. And what they will do? If a pathogen is entering into our body, now these cells, what they are doing? They are trying to engulf, E-N-G-U-L-F, engulf, E-N-G-U-L-F, engulf means what? Eating like this. So they are engulfing it, they are eating it, so that inside the cell now it will be digested, it will be broken. So in this way, they are involving in protection from the pathogens. In this way, neutrophils and monocytes, both of them are phagocytic in function. In this way, they are trying to protect our body. Okay, both of them functions completed. Now let us see about the lymphocytes. What these lymphocytes? Lymphocytes are two types: B lymphocytes, T lymphocytes. B lymphocytes and T lymphocytes. B lymphocytes will produce antibodies. Antibodies, they are also called as immunoglobulins. In the previous lecture, I told you about this. Antibodies are also called as immunoglobulins. T lymphocytes, B lymphocytes. Ultimately, what is the function of this? Exact mechanism I will not explain here because we will study under health and diseases. B and T lymphocytes, they are helping in fighting with the antigens, fighting with the pathogens, so that they are protecting us from so many infections. Clear? All these are whether directly or indirectly, ultimately they are helping us in protection from various pathogens. This is what the general basic idea regarding this. In the NCERT they have given percentage also. Most abundant are neutrophils and least abundant are basophils. Number wise if you see. This is what the one important point. Clear? So these are the other important functions of these particular cells, and here I have given the list. Neutrophils are the most abundant, 60 to 65% are present, and these are most abundant cells, and these are phagocytic cells. How they are helping in protection from the pathogens? By doing the phagocytosis. They are engulfing the pathogens and then they are protecting us, and which destroy the foreign organisms. Basophils, these are the least abundant, just 0.5 to 1% only, and they are secreting different chemicals like histamines, serotonin and heparin, and generally they are involved in inflammatory reactions. Okay, next one. But heparin is anticoagulant, so please remember this. Eosinophils, they are approximately 2 to 3%, and they are resisting, they are protecting us from so many infections, and generally they are associated with allergic reactions, and they are also involved in some parasitic, helminthic parasitic infections. Okay, next one. Lymphocytes, B lymphocytes, T lymphocytes we are studying, and they are 20 to 25%, and they are generally involving in immune response of the body. Their role is something very great, very great. Those things separately we will study under health and diseases chapter. Next one is regarding monocytes, approximately 6 to 8%, and they are also phagocytic cells, and this way they are destroying the organisms. So abundance wise if you see, most abundant are neutrophils. After this, lymphocytes. After this, monocytes. After this, eosinophils. After this, basophils. Clear? This is what the sequence wise if they ask. So this is what the point is. Now let us quickly go through NCERT content. Leukocytes are also known as white blood cells, WBCs, as they are colorless because they are not having the hemoglobin due to the lack. Lack means what? Absence of hemoglobin. They are nucleated cells. RBCs, which is RBCs are not having nucleus, but these are having the nucleus and are relatively lesser in number. So their number is very less, and average number is 6,000 to 8,000 per cubic millimeter of the blood. Next one. Leukocytes are generally short-lived. Their lifespan is very less. And we have two main categories of WBCs. They are called granulocytes and agranulocytes. What are the granulocytes? Neutrophils, eosinophils and basophils are the different types of granulocytes, while lymphocytes and monocytes are the types of agranulocytes. In this way, two groups are present. Neutrophils are most abundant. Percentage is 60 to 65% of the total RBC, WBCs, and basophils are the least abundant, just 0.5 to 1% among them, among the total RBCs, total WBCs. Next one. Neutrophils and monocytes, both of them. Monocytes percentage is 6 to 8%. Neutrophils and monocytes are phagocytic cells. They are destroying the pathogens by phagocytosis, and thereby they are destroying the foreign organisms that are entering into the body. So in this way they are giving protection. Basophils will secrete histamine, serotonin, heparin, etc., which are involved in inflammatory reactions. But please remember, histamine, serotonin are involved in inflammation, but heparin is anticoagulant. It will prevent the coagulation or clotting of the blood that is flowing in the blood vessels. Okay, next one. Eosinophils are just 2 to 3%. They are resisting, they are protecting us from infections and are also associated with allergic reactions. And they are mainly protecting from infections. Which infections? Mainly parasitic like helminthic infections. Next one is regarding lymphocytes. They are 20 to 25%, and these are of two major types, B lymphocytes and T lymphocytes. Within the T lymphocytes, again different types are there, TH cells, TC cells, likewise many are there. Now we don't need to worry about them. So in that particular health and diseases chapter we will study. Okay, next one. Both B and T lymphocytes are responsible for immune functions of the body. So they are having very major role in the immune system. So those things we will study separately. Next one is regarding thrombocytes. They are also called as platelets. And here, what is the normal count of this? Already we studied, no? 1,50,000 to 3,50,000. So that is the normal count for these particular platelets. And now the production of this particular platelets is called as thrombopoiesis. And what is the shape of these? These are disc-like. But what about the color? These are not having hemoglobin. That's why these are also colorless. And next one is regarding the lifespan. What is the lifespan of this? Their lifespan is approximately 7 to 10 days. That's it. 7 to 10 days is the approximate lifespan. But what about the nucleus? Here I have not written about this, but what about the nucleus? This point is very important. Observe carefully. RBCs, mature RBCs do not contain nucleus. Mature RBCs, RBC. Regarding the nucleus, please observe carefully. RBC does not contain nucleus. WBCs, all the 5 types of WBCs, yes, they have the nucleus. But what about the platelets? What about the platelets? Platelets does not contain the nucleus because actually they are not true cells. Even though you are saying that they are cells, but actually they are not true cells. They are not true cells. Why you are saying like this? Because within the bone marrow, there is a cell that is called as megakaryocyte cell. Megakaryocyte cell. Please observe carefully. Within the bone marrow, there is a cell called as megakaryocyte cell, and this megakaryocyte cell will break into so many small fragments, so that now they are forming some small disc-like structures. And these cell fragments, these fragments of the cell are called as platelets. So that's why platelets are actually not the complete cells. Please observe carefully. Platelets are not complete cells. Actually, the cell called megakaryocyte cell is broken into so many small fragments, and these fragments are called as platelets. Since they are the cell fragments, they are not the complete cells, they don't have the nucleus. Please observe carefully here. So they are the fragments of megakaryocyte cell. They are not complete cells. They don't have the nucleus. This is the most important point you need to understand. Okay. And next important point I am linking with other chapter is, so in the genetics there is a chapter called as molecular base of inheritance. In that chapter at the end we will study about DNA fingerprinting. So at least you heard about that word, whether you know the process or not, no problem, but at least you know about a particular name, DNA fingerprinting. In the DNA fingerprinting, first you need to get DNA from an individual. If you are getting an DNA from individual, what is the best source of DNA? Blood collection. Suppose you want to get DNA from my sample, then what is the best sample? Blood. Simply take a small amount of the blood. From the blood, take the cells. From the cells, take the DNA. Over. But what is the technical question here? You are isolating blood, okay. And from the blood, in the blood there are 3 types of cells, fine. And you are isolating DNA from the cells, that is also fine. If you are isolating DNA from the blood cells means technically you are isolating from which cells? You are isolating from the WBCs, because only WBCs are having the nucleus. From the blood you are isolating DNA means what? You are isolating DNA from WBC, because mature RBC does not contain nucleus, and even platelets are also not containing the nucleus. From the blood you are isolating DNA means you are isolating DNA from WBCs. This is a very important point. You need to remember this. This point once again we will discuss when we are studying that DNA fingerprinting concept in the genetics. Please remember this. So in this way, what is the ultimate content? Platelets are also not having the nucleus. They are small disc-like cell fragments actually. So this is what the point is. Clear? What is their function? These platelets will release some components which are involving in blood clotting. So anyhow we will study about coagulation of blood or blood clotting. At that time once again you will study its role. So platelets are releasing some compounds which are involving in blood clotting. So for blood clotting or coagulation, these are very important. What happens if their number is decreased? even for a small cut, we will lose so much of blood, because if their count is less, that blood coagulation initiation should be done by these chemicals. If these chemicals are not produced, now who will initiate this clotting? Someone has to tell that you should start the process of clotting. And if these chemicals are not present, now what happens? Clotting will not occur. So even for small cut also we will lose so much of the blood. So this is what the problem is. So that's why these cells should be present within the normal range. So that's what the significance of this particular cell types. Okay. Platelets are also called as thrombocytes, and actually they are cell fragments. Better underline this particular one. They are not the complete cells. They are actually cell fragments that are produced from megakaryocytes. Underline this one also. Megakaryocyte cell. And these are the specialized cells that are present in bone marrow. Next one, blood normally contains average value. These commas they have given wrong, so observe carefully. 1,50,000. Okay. If you want to keep as after 3 zeros, now it will be 150,000. Likewise, you need to say. You can say this as 150,000 to 350,000, or else in our version, if you are saying 1,50,000 to 3,50,000. So with respect to these commas, there is a mistake. Observe carefully. And they are having 1,50,000 to 3,50,000 platelets per cubic millimeter. Platelets can release a variety of substances, most of which are involved in coagulation or clotting of the blood. That's why there once again we will study. A reduction in their number, so reduction in the number of platelets can lead to clotting disorders. If their number is decreased, now clotting does not occur, so clotting disorders will occur, and which will lead to excess loss of blood from the body. For whenever there are cuts, if the platelet count is less decreased, now what happens? So huge loss of blood will occur. This is what the problem is. So in this way, we are studying about these formed elements in this particular lecture. So with this, introduction is completed. So there individually for plasma we have completed, and individually the functions of these cell types also we have completed. Clear? With this particular part is completed. So in the next coming lectures we will go with remaining other concepts. Okay, we will meet in the next lecture. Thank you.